Now, I'm going to ask someone whom I'm going to ask. Or go for that. Tell me actually, did I complete this thing?
Sir,
Or Prabhu, do you hear me?
yes, sir.
আচ্ছা কিংডম মারা কমপ্লিট হয়ে গেছিল
Sir, bacteriophage life cycle পর্যন্ত complete করেছি।
হ্যাঁ হ্যাঁ বলো
ব্যাকটেরিওফাজের লাইফ সাইকেল পর্যন্ত শেষ।
আচ্ছা আর কি ব্যাকটেরিয়া সায়ানোব্যাকটেরিয়া হয়েছিল
হ্যাঁ হ্যাঁ ওগুলো
হয়েছিল না ব্যাকটেরিয়াল ক্লাসিফিকেশন আচ্ছা আই এম জাস্ট রাইট ইউ জাস্ট টেল মি
স্যার ব্যাকটেরিয়াল নিউট্রিশন হয়নি।
আচ্ছা ঠিক আছে ক্লাসিফিকেশন অফ ব্যাকটেরিয়া based on mode of nutrition. Okay. Now you see here, you can classify it like this. Autotrophs, heterotrophs. Chemoautotrophs. Autotrophs can photosynthesize and so their reaction will be like this: rho 6CO2 plus 12H2S sunlight bacteriochlorophyll plus the example, a lot of examples are there, like chlorobium. So they actually photosynthesize with the help of sunlight and bacteriochlorophyll. Then they are the photoautotrophs. Those who are chemoautotrophs, they are to derive nutrition. Chemoautotrophs are to derive nutrition from inorganic compounds. Then you can call them chemoautotrophs. They derive nutrition from inorganic compounds. Then you can call them chemoautotrophs. So, example, Nitrobacter. Nitrobacter is a bacterium. It helps in the conversion of nitrite to nitrate. It helps in the conversion of nitrite to nitrate. Then you can call them the Nitrobacter. And it is actually a kind of chemoautotroph. That means it derives nutrition from inorganic compounds. And there are some sulfur bacteria, nitrifying bacteria, they all are chemoautotrophs and hence they help or they play an important role in recycling of nutrition. Remember this thing, they play an important role in the recycling of nutrients like nitrogen, phosphorus, etc. So, this is what about the chemoautotrophs. Already I give you the example. Then comes to the heterotrophs. Is it clear up to this? Is it clear up to this that who are autotrophs? When you present photoautotrophs and what is the meaning of the chemoautotrophs? I think it is clear to you. Now, you see, you remember this thing is a very most important question in terms of entrance. They play an important role in the recycling of nutrients. Remember this thing. Who? The chemoautotrophs play an important role in recycling of nutrients, different nutrients. Regarding heterotrophs now. Now I am going to show you about the heterotrophs. Heterotrophs can be classified in this way. I am going to rub it out. Heterotrophic bacteria saprophytic bacteria symbiotic bacteria. So heterotrophic bacteria of three types, saprophyte. Who are saprophyte? Remember this thing, the organisms which derive nutrition from dead and decaying matter. They derive nutrition from dead and decaying matter, then you can call them saprophytic bacteria. Example, Pseudomonas. Pseudomonas is a saprophytic bacteria. You know, whenever any kind of petroleum leak occurs in the sea, then this bacterium helps in splitting the oil pills. So it breaks down the petrol. So these are the saprophytic bacteria. Saprophyte is not the only bacteria saprophyte. A lot of organisms under the kingdom fungi are found to become saprophytic in nature. So whenever the term saprophyte is used, remember this thing, they have to derive nutrition always from dead and decaying matter. Comes to the parasitic bacteria. Before that, I would like to ask you about the parasite. I'm going to ask, Urpita Bhomik, do you hear? Parasite.
Sir, parasite is a host that lives in a living organism.
Please, repeat again. Parasite is a
Sir, it's a host or a different kind of organism which lives in other organisms and takes nutrition from that organism.
Okay. Anyone else to add something? Somujit Das. What is a parasite?
The organisms which obtain their food and shelter from other organisms without harming themselves are called parasites.
Without harming. Okay, Orko Prabhu, you raise your hand. Orko Prabhu.
Sir, parasites are some organisms which depend on a host
A bit louder, please. A bit louder.
Sir, parasites are organisms which depend on a host organism for their nutrition and shelter.
Female Anopheles or female mosquitoes. You know, female mosquitoes are actually sucking blood from us. Now, if I am going to ask whether female mosquitoes are parasites, what should be your answer?
Yes, sir.
Female yes,
Yes, sir.
tell me.
They are ectoparasites.
That means you are telling that female mosquitoes are parasites as they are sucking blood from us. Okay. Now, I am going to write. I am not going to answer this question directly. Rather, I am going to write something here regarding parasites. You know, in order to be considered as parasites, in order to be considered as parasites, following two conditions are required to be satisfied. What is written? In order to be considered as parasite, following two conditions are required to be satisfied. What are these two conditions? I am writing around. The parasite is to draw nutrition from host. It is to draw nutrition from host. First thing. Second thing, in host parasite interaction, the host is required to be harmed. Two statements are given here. One is that the parasite is to draw nutrition from hosts, and secondly, the host parasite interaction, the host is required to be harmed. Are you getting me? In a host parasite interaction, the host is required to be harmed. So this is what about the parasite. If it is so, if these two conditions are required to be satisfied, then think about the female mosquito. Female mosquito is sucking blood from us. This is true. But the amount of blood sucked by mosquito, whether this amount can do harm to us, if mosquitoes are biting throughout the day even, how much blood can be sucked by them? 10 ml, 15 ml? But in a blood donation camp, we used to donate 300 ml of blood. That means the amount of blood sucked by mosquito from us, these amounts, if it is even withdrawn, taken away, we will not be affected at all. That's why, in terms of mosquito, this condition cannot be satisfied. So we are not harmed at all. due to sucking of blood by mosquitoes. It's a different thing. If plasmodium is there, then it will cause malaria, but it is not the mosquito. Plasmodium causes malaria. Plasmodium is a parasite. This is true. But female mosquitoes should not be considered as parasites as although they pass blood. So one condition is satisfied, but the other condition is not satisfied. That's why female mosquitoes are not considered as parasite actually. Because although the first condition that is they are deriving nutrition from us, obviously, this is true. But in host parasite interaction, when interaction between human and mosquito, the amount of blood sucked by mosquito is not going to do any harm to human being. That's why the mosquitoes, female mosquitoes are not considered as parasite in this regard. Are you getting me? Is it okay? Okay.
Yes, sir.
Yes, sir.
Yes, sir.
Now, okay. Now we are going to now regarding the similar thing, the parasitic bacteria. That we are discussing about the parasitic bacteria. Now you see a lot of bacteria there. Known as the disease-causing bacteria. Suppose cholera, typhoid, tuberculosis, tetanus, a lot of them. Now, you need to understand, you have to remember the name, Vibrio, cholera,
Salmonella, typhoid,
typhi,
Mycobacterium, tuberculosis,
Clostridium tetani. A lot of that. Suppose in case of suppose some of diphtheria is there, Furinobacterium diphtheria. Suppose plague is there, Mycobacterium leprae, a lot of there. So you need to understand. Now write down one question, please. Write 4, please, everyone. Write 4 human diseases. Or write 6 here. Put to there. Write 6 human diseases caused by bacteria. Write 6 human diseases caused by bacteria. And write down the name of causal organism in each case. And write down the name of causal organism in each case.
Sir, can you please repeat the second part?
Okay, I am repeating the whole. Write 4 human diseases. Write 4 human diseases caused by bacteria. And write down the name of causal organism. That is the bacteria that causes the disease. Name of the causal organism in each case. Okay? Next slide. Next question. Write 2 plant diseases. Write 2 plant diseases caused by bacteria. And write the name of causal organism in each case. Okay? Next, write 4 beneficial bacteria.
Sir, please repeat.
Write 4 beneficial bacteria. Same question, how are they beneficial? Okay? Now I am going to the third type under autotrophic bacteria. Third type is symbiotic bacteria. I think you are aware of this term symbiosis. Symbiotic bacteria are those bacteria who are found to be in symbiotic association with the other. What is meant by symbiotic association? Symbiotic association is a type of association between two organisms of different species. It is an association between two organisms of different species who are lying together and nutritionally benefited. For example, I am telling one example, Rhizobium is a soil bacterium. And the other one, any legume or leguminous plants, like soybean, like different types of lentils, etc. Now there is an association between these two. In the roots of leguminous plants, if it is a leguminous plant, and inside this root nodule, rhizobium is found. Inside the type of association, rhizobium is found is present inside the nodule. It has in the fixation of nitrogen, so that plant is benefited. They are getting nitrogen nutrition. In the rhizobium is also getting nutrition from the plant. In this way, both are mutually benefited. Actually, in this association, both the organisms are mutually benefited. In this case, both the organisms are mutually benefited. Then you can use the term symbiosis. For example, I am giving another example here. You will get another bacterium called Frankia. Example 1, example 2. Frankia, another bacterium. Found in the root nodule of Elnas tree. Here again, There is an association between Frankia and Atlas. Atlas is a non-leguminous plant. It is an exception. Mostly rhizomia is found in leguminous plants. So Frankia is another type of nitrogen-fixing bacteria found in the nodules of Atlas. So these are the symbiotic association. You need to remember one thing. Whenever it is a symbiotic association, every time due to this symbiotic association, both are the organisms which are involved in this association. They should belong to two different species and they are required to be mutually benefited. Otherwise, you cannot call it symbiotic association. And how they are benefited? And this benefit in terms of nutrition. They should be nutritionally mutually benefited. Otherwise, you cannot consider it symbiotic association. So this is called symbiotic association and they are the symbiotic bacteria. Rhizobium, as an example I gave you, this is the Frankia. I think it is clear to you now about the symbiotic bacteria. Another example from the other part, I think you heard about the lichens? Aparajita Devdas, do you hear me? Aparajita, do you hear me? Algal part is called phycobions and fungal part is called mycobions. Myco comes from mycology, that is the study of fungi. From here, myco comes. Phycobion, the fungal part, so algal part, the algae, you know, the study of algae called phycology. From here, the term comes myco. What is about this is the actually association. In this association, the algae can prepare food for which different inorganic nutrients and water can be supplied by the fungi. Once food is prepared, a part of which is taken out by the fungal part. So this is, you know, some you can get some patches on some tree, like, you know, different types of tree, you know, suppose gray bluish patches are found on the bark of some tree, and these are the lichens. Okay, I will be showing you, I will be showing you someday that what are lichens actually. You will get it. In different trees, some patches are found, like that if it is a tree, like this, you will get, suppose patches over here. These are patches. A grayish blue patches found, and these are nothing but the lichens. You will get it. If you observe the trees around you, you will get it somehow, I think. And lichen is a pollution indicator also. You know, lichen is a indicator. What type of pollution? Mostly the sulfur dioxide pollution. That means sensitive to sulfur dioxide. If the concentration of sulfur dioxide due to industrial pollution increases, then you will see that the lichens are actually washed out and they will become dead. So where there is huge industrial pollution, there is huge emission of sulfur dioxide somehow, then there will be no lichens. In our state, there is no such pollution actually, so you can get lichens everywhere. And there is a actually story about the some butterflies, moths, actually, you know, Biston Bitularia in England. At that time, before industrial pollution, before the industrial revolution, there were huge number of lichens there, and some butterfly called Biston Bitularia, the two sets, one was white, other is blue, other is actually black. The white one can easily camouflage there. White one can easily camouflage there, and So that what will happen, the white one's number increases. But because the same predator bird used to eat both white one and black one, the white moth and black moth. So what happens as the pollution is very clean, sorry, environment is very clean, there is no pollution because it is before the industrial revolution. So there is huge amount of lichen there and white moth can easily camouflage in white lichen so that they cannot be traced by the bird. So their number increases, but the black one cannot camouflage. So their number gradually in England, there was huge emission of sulfur dioxide. And due to huge emission of sulfur dioxide, as lichen is very sensitive to sulfur dioxide, all these lichens are killed. Now it is very difficult for the white moth to protect themselves because they cannot camouflage for that. But as environment is dark now due to pollution, the black moth now can easily camouflage in this environment. And due to which the number of black moths gradually increasing and number of white moths gradually decreasing. You will get it in class 12 again. So this is about the lichen because why lichens were destroyed at that time? Due to huge amount of sulfur dioxide in the atmosphere. The lichen cannot tolerate sulfur dioxide and hence they are killed. Are you getting me? So this is what about
স্যার
বলো বলো
স্যার ওই ফাঙ্গাল পার্টে কি লেখা আছে এটা দেখা যাচ্ছে না।
ফাঙ্গাল পার্ট ওকে microbiome. Why microbiome? Because microbiome is the study of fungus. হ্যাঁ বলো
ব্ল্যাক মথ কি পলিউটেড এনভায়রনমেন্টে ক্যামোফ্লেজ করতে পারে?
না দেখো, if the environment is polluted, then what is the basic thing here? Pollution increases darkness. There will be huge dark. And gradually the environment is around it becomes to some extent black. Then you see, if you go to the slum areas regarding roadside, you will get a grayish black covering everywhere. Have you seen this?
Yes, sir.
So the covering is black, actually or dark. Gradually due to pollution, there will be increase in darkness, blackness. So the black one can easily camouflage in this environment. But white one is easily visible now. It cannot camouflage. And that's why it is easily traced by the bird who is eating both black and white moth. Now black moth cannot be visible. White moths are visible, their number gradually decreasing. You will get it in class 12, it's a kind of directional selection. Selection is directed towards the black from the white after industrial revolution. Getting me now?
Yes, sir.
Okay. Now, so this is all about the, now another classification is required here. Bacteria based on the mode of respiration. Bacteria aerobic, facultative, obligate.
2808. Zero and 0 0 and 69 and 2 and
I was discussing that of the facultative, the aerobic and anaerobic facultative again, and obligate. What about the term obligate is used? Obligate means compulsorily it should be aerobic. Compulsorily aerobic means it cannot survive in absence of oxygen. However, it is compulsorily aerobic, that it cannot survive in absence of oxygen, like Bacillus subtilis. Normally anaerobic. They are normally anaerobic, but when oxygen is available, they can switch over to aerobic. That means normally the facultative one, that means aerobic is also facultative and obligate. Those who are facultative, they, normally they are anaerobic bacteria. Facultative aerobic means they are normally anaerobic bacteria, but wherever oxygen is available in the medium, they can switch over to become aerobic bacteria. Then they are called facultative aerob. So Rhodopseudomonas. Pseudomonas. Then regarding anaerobic. Again, two terms. One is obligate part. Again, they can survive only when oxygen is not available. That means they can survive only in absence of oxygen. Then they are called obligate anaerobic. That means for their survival, an anaerobic condition is required to be maintained. If somehow the condition cannot be maintained, then obviously they will. What will happen? Obviously they will die. Example, Clostridium. Tetani. Clostridium tetani is actually the obligate anaerobic organism. That means if oxygen is not available, then Clostridium tetani cannot survive. Next one, facultative. Facultative is again normally facultative anaerobic are normally aerobic. Normally they are aerobic, but whenever oxygen is not available, they can be converted to aerobic. Are you getting me? Facultative anaerobic organisms or bacteria, normally they are aerobic bacteria. They are normally aerobic bacteria. They can survive in the presence of oxygen. But when oxygen is not available, they become what? They become anaerobic. They are called halophils. So these are the classification of bacteria based on the mode of nutrition, mode of respiration. Sorry. Now, one thing is left, so I recall. This is regarding, I think I completed the virus. I forgot about the viroids, prions. These two things may be left. I think I discussed, I forget it. Okay, simply I am telling you, in terms of viroid, actually not a virus. They are the, both are cellular entities. But viroid is not a virus. Viroid actually viroid. Avadar, do you hear me? Avadar, do you hear me?
Yes, sir.
Tell me, what is capsid? A bit louder.
Sir, can you please repeat?
What is louder, louder? I'm not hearing you. Louder. Vishakhapatnam. Okay. Also, Prabhu,
Yes, sir.
you tell me what is capsid?
Sir, I don't remember.
What is capsid? Okay, also, you raise your hand. Tell me.
Sir, the protein code of virus is called capsid.
Now it is very clear, clearly audible, Autonomous hand. Autonomous, from where you are calling from? Kamalpur.
Yes, sir.
Okay, very clear, but otherwise voice is not coming. Perhaps he is far away from the mic. Or he has some problem with his mic. Okay, it's very good. The capsid is the protein code covering the nucleus. Basically, in order to become virus, a capsid is required to be present as virus and is also having low molecular weight. Other terms comes the pre.
না দেরি করছে আসতে শোনা যায় না।
Tiny, synthesized is called primary protein. Then comes to the secondary protein. Then comes to tertiary protein. You see, during this transformation from primary to secondary to tertiary, there is folding. Folding is going on. And during this folding, some more bonds are developed. If somehow this folding is not a proper folding, or due to misfolding, some prions are developed. Actually, prions are, you can call misfolded protein. They are the misfolded protein, which are not properly folded, misfolded protein. And these misfolded proteins are actually these proteins which are not properly folded. So, and in the host, suppose this is a host, if suppose this protein enters, suppose this is the protein, so this is the prion. And what enters inside the host, system, and however these proteins have some actually misfolded, is actually affected. I will be showing you one video next day showing that how prions are actually completing its life cycle. So this is what about the prions. We have these two things. One is actually now write down one. Just write one question. What are prions? How are prions different from viruses? Oblique virus. Name one human disease caused by prions. Caused by prions. Okay. So you have to go. There is a new book also. Name one human and one category also you can write. So this is what about the prions, and in this way the kingdom Monera is completed now. We have to proceed towards the next kingdom now. Kingdom Pratishtha. So what's the basic thing? Eukaryotic. That means they all are eukaryotic. All the kingdom Monera are portrayed. One thing you note it down, please. As I'm writing, that is maximum nutritional diversity is found in kingdom Monera. Maximum nutritional diversity is found in kingdom Monera. Sometimes this is the most important question coming in MCQ test. Sommajit Das, do you have any question?
Sir, what is the relation of prion and kingdom? What is the relation of prion and kingdom Monera? Sir, what is the relation of prion and kingdom Monera?
Look one thing. Prions, virus, they are not included in kingdom Monera. Actually, in even since kingdom of life, there is no space for all these acellular entities. Remember this thing? Obviously, you need to remember it that kingdom Monera doesn't include virus, and the prions. Basically, acellular organisms are not at all included in any kingdom, in any domain. Are you getting me now?
Yes, sir.
Now you see, regarding kingdom Pratishtha, first thing, they are all eukaryotic in nature. Secondly, they are unicellular. They all unicellular organisms, all unicellular eukaryotic organisms are now included under kingdom Pratishtha. Are now under kingdom Pratishtha. So if you are going to classify it, Pratishtha algae, slime molds, and protozoan protist. Pratishtha algae, slime molds, and protozoan protist. Look one thing, Pratishtha algae. In case of Pratishtha algae, you see, we have C1. One is dinoflagellate. Two, chrysophytes. Three, euglenoid. Suppose, for example, Noctiluca. Suppose actually, you know, what is the diatoms? You see, there are two types. I'll explain them later on. Again, golden brown and golden diatom. Then comes to the plant modes are of two types. Acellular cellular. Protozoan protists are of two types. It is, you know, Sarcodina Sporozoa, ciliata. There are three types. Now, Sarcodina includes, suppose amoeba, you know, it includes Plasmodium, it's the Paramecium.
I can't see. It is not visible.
Okay. Cellular, cellular. Sarcodina, Sporozoa, under them, Sarcodina you will get amoeba. Under this you will get the Plasmodium. Another one is left, the Jugflagellata. Under these Jugflagellata, under this you will get suppose Trypanosoma. Trypanosoma. So you see Sarcodina, amoeba, Jugflagellata, Trypanosoma, Sporozoa, Plasmodium, ciliata, Paramecium. So this is the group we have to study now. What are they? Dinoflagellates, Crassifiers and Euglenias, they are one group, they are the protista algae. Under slime mode, you will get acellular and cellular. And slime modes are actually called fungi animal. I will be discussing this later on. Protozoan protist includes Jugflagellata, like Trypanosoma, what is called Leishmania. Sarcodina includes amoeba, Sporozoa includes Plasmodium, and ciliata includes Paramecium. So you have to start from the protista algae first. So you have to see the basic structure, you need to draw it, and you have to start from the protista algae first. So you have to start from protista algae. I have to write it out now. Yes.
Sir, Obama messaged me that he is in the waiting room. Obama messaged me that he is in the waiting room.
Okay. Now you see, now dinoflagellate. We have to start from here. First thing, having flagella. Sometimes Indian can flagella mostly is heterocon type. Mostly it is heterocon type. Heterocon type means when two flagella are found to be equal in all respects, then you can call them homocons. And if they are found to be position to different parts, one is longitudinal, other is lateral, or unequal in size, then the term used is heterocon type. And in this case, flagella heterocon type first thing. Remember this thing, flagella are heterocon type. Second thing, what about the nutrition? Yes.
Sir, please explain the heterocon type again.
Look one thing, I am starting from homocont again. Whenever the flagella are found to be with similar size and shape like this, you see, this is one type. Or if flagella appear like this, look here. So this type heterocon flagellation. Here it is homocont. Getting now? Clear to you now? What is heterocon?
Yes, sir. Yes, sir.
Now, yes.
Sir, what is the difference between heterocont and homocont?
What is that?
So what is the difference between
Yes.
heterocont and homocont?
And homocont.
Yes, sir.
In heterocont, and
Sir, the difference
what is the
between
difference between?
the difference between homocont and heterocont.
In homocont, both the flagella are found to be equal in size and shape, and they are produced from the same point of origin. But in case of heterocont, they may be of different point of origin and they are not equal in size and shape. Are you getting me now, Orko Prabhu?
Yes, sir. Yes, sir.
Okay. Now, regarding the other point, you see, flagella is there, okay, having flagella, that's why dinoflagella is also the name. Just a minute. Go. No, just a minute here. First thing is having flagella, heterocon type, dinoflagellate. Second thing, there may be autotrophs. But exception is there. So Noctiluca shows holozoic nutrition. Noctiluca shows holozoic nutrition. Normally, Goniaulus. Exception Noctiluca, although it is under the dinoflagellate, but it shows holozoic nutrition. Okay? Another most important property shown by they show the property of they show
So sir, generally they are autotrophs.
huh?
So sir, generally they are autotrophs.
Autotrophs. But some exception is the octulus is bound to be autotroph. You know, protistan algae mostly autotroph.
What is
Okay.
holozoic nutrition?
Holozoic means when it derives nutrition from other organisms. Sketching organisms like us, we are showing holozoic nutrition. What the example of holozoic nutrition? Are you getting me? Bioluminescence, what does it mean? I think you can see the firefly. Light is emitting out from them. Have you ever seen them? Firefly?
Yes, sir.
Yes, sir.
Okay. So, what is the source of this light? How is light produced actually? What is happening in them? Basically, you know, the organisms which are showing bioluminescence, that a specific compound called luciferin. A specific compound called luciferin is found, and luciferin along with other enzyme, luciferase. That means in order to emit light, if someone is to emit light, then they should have this luciferin and also the luciferase. Generally, ATP is generated. Now, what happens here? Initially, once ATP is generated, luciferin binds to ATP. What is happening actually? Luciferin binds to ATP. Conforms luciferin ATP compound. First of all, I am repeating it again. Luciferin, the pigment present in these organisms showing bioluminescence. This luciferin is found to, what is going to do? Luciferin is binding with that of ATP produced in aerobic respiration. Then this ATP, luciferin ATP compound is broken down or oxidized by the luciferase, so that some energy of ATP is released out as light energy. Some energy of ATP is released out as what I say, light energy. ATP, you know, there are 3 phosphate groups, but one phosphate group is released out as light energy. And so remember this thing, in order to become bioluminescent, you know, bioluminescence is found in different kingdoms. So in order to show bioluminescence, 2 things most important. One is luciferin is required to be present, and secondly, luciferase enzyme is required to be present. If luciferin and luciferase both are not found, obviously the organism cannot show bioluminescent property. I repeat again, luciferin is a pigment present in the bioluminescent organism which is to bind to the ATP, forming luciferin ATP compound. Then in the presence of what it is called the luciferase enzyme, the luciferin gets oxidized, and from ATP, one phosphate group is released out as light energy. And this is the light coming out even in case of firefly. And you know, in firefly also, there is what? In firefly also, you will get the luciferin and luciferase. Now today I am going to stop now. Our next class will be on next Sunday, I think, at 8 or 8:10 a.m. Thank you very much. Have a good day. If you have any problems,